important thing is not to stop questioning.
Yes, that's important. Please ask as many questions as you possibly can. And I'm not getting many. I think it would help you if you would ask questions. So real quick, I want to go over respiratory pigments. Um, the whole idea behind a respiratory pigment is that oxygen is not very soluble in water. Fish have the countercurrent exchange mechanism to get as much oxygen as they can out of the water. However, and they need that countercurrent exchange. However, oxygen does not typically dissolve in water at high concentrations. And if it can't dissolve in water at high concentrations, it couldn't dissolve in the blood at high concentrations. So to solve this, animals use respiratory pigments, and they use different respiratory pigments. I've got them listed here. One is called hemocyanin. This is the copper-based ones that you see. You've heard of like the horseshoe crab, the arthropods having blue blood, and it does appear blue because their respiratory pigment molecule has a color, and it's to help capture oxygen that wouldn't normally dissolve in the liquid. Their hemocyanin is copper-based. Ours is called hemoglobin. Here's our hemoglobin. We've seen this a bunch of times. Flashback from protein quaternary structure. This is a polypeptide that has gone through secondary, the beta pleated sheets and the alpha helices, and then the tertiary folding with the disulfide bridges. We have a prosthetic group here. This is a porphyrin ring, and on the inside of that is an actual iron atom. That's why iron is important. The whole ring itself is called heme. That's where the hemoglobin comes from. We have two beta subunits, two alpha subunits together. It makes up the quaternary structure of a protein. So this is where we have seen this. It just keeps coming back because hemoglobin is pretty important. What happens is each one of these heme groups, or each iron atom, attaches to an oxygen, an O2. So diatomic oxygen molecule. It can carry an O2 or a diatomic oxygen molecule, and that increases the solubility of oxygen in the blood because for every red blood cell, and this is only red blood cells that have it, so each RBC has about 250,000 copies of hemoglobin, which really increases the amount of oxygen that can be transported in the blood. Now, the key here, there are two key components to this. Each one of these binds, but it's the first one that's really key. The first one, whichever first one it is, it puckers this heme ring, this porphyrin ring. It puckers it in a way that pulls on this structure, that pulls on the hydrogen bonds of this structure, that pulls on the hydrogen bonds of this structure, that pulls on the hydrogen bonds of this structure, and it causes what's called cooperativity. Once one oxygen binds, it actually puckers the other rings out to make it even easier for the other three to bind. So as soon as you have one to bind, the other three bind even easier. That's the idea of cooperativity, and that is critical. One, these guys are moving fairly quickly, but to facilitate the uptake of high oxygen concentrations in the lung into the blood. So as soon as one changes. Now the other neat thing is then if you put this hemoglobin molecule in a low pH, those hydrogen ions in solution interact with R groups on the surface, and it actually breaks those little connections and it shifts back to another shape. It shifts in a way that it actually pops out the oxygen. So in a low pH, these oxygens are popped out easily. and more easily than diffusion would have them just come out from a high concentration to a low concentration. Now, when would hemoglobin find an area of low pH? Well, remember, carbon dioxide in the plasma plus water makes HCO3, carbonic acid, right? So we get the carbonic acid lowers the pH. So if you have a tissue that the mitochondria is bringing in a whole bunch of oxygen, letting out a whole bunch of CO2, the pH around that tissue drops. The hemoglobin going by the interstitial fluid hits an area of low pH. The shape of the hemoglobin itself changes. The shape of the hemoglobin changes. It pops out the oxygen. Now it's in a conformation. It's in a shape that will actually very easily either pick up hydrogen ions, and with that, the bicarbonate ion will follow, or it can actually also pick up the CO2, but the conformation changes, which helps. Now, the last one is called myoglobin. And this guy is a really neat respiratory pigment. You see this in diving mammals, diving sea mammals. Sea mammals. I don't know of any other diving mammals but in the sea, but diving mammals. And some seals and Weddell seals, I know, whales, they have myoglobin, which is a respiratory pigment in their muscles, and it will hold on to oxygen in their muscles. So when they are down and diving down and they stay down for 20 minutes, you go, how can they? How can they possibly stay down that long? It's because this respiratory pigment pulls the oxygen out of the blood and stores it in the muscle tissue. And then depending on how long they're down there or how much exercise they're doing when they're down there, which isn't much because they're that fusiform shape, so they don't have much drag. They can take it nice and slow. The oxygen actually is used out of their muscles. And unlike us, well, we kind of can, they can ferment if they have to. They can undergo a fermentation process if they have to in their muscles. Now, we go through a lactic acid production in our muscles, but they have a way of not getting sore if they go to this non-oxygen environment, but they really don't have to because they store so much of their oxygen in their muscles and tissues, and that's how they can hold their breath for such a long period of time. So that's where the myoglobin respiratory pigment comes from. And I think we're summing it up. Let's see, did I forget anything? Put in another thing. Okay, so just to review, cooperativity is the conformational change to the heme molecule. And do you remember what causes it? Due to the binding of one O2, and it increases the affinity, which increases the affinity of the other free O2. Now the Bohr shift. Okay, the Bohr shift. This is the pH. So a low pH causes the release of O2. So your book shows you a graph, and it shows you the normal release of oxygen from hemoglobin, the normal rate in the blood. However, if you lower the pH, that rate increases. The difference here is called the Bohr shift. So a low pH causes the release of O2. So when your blood hits a tissue with a lower pH, then it is more likely to drop off the oxygen. It actually releases the oxygen at a greater rate than in a tissue that doesn't have that low pH. And that is called the Bohr shift. So low pH causes the release of oxygen from heme. And the way this does it, low pH causes the release. It also causes the conformational change. So it's all back to the shape of a protein. Conformational change. Cooperativity and Bohr shift. Good luck.